So we're going to continue with our talk about the cell cycle. So on the last lecture, I talked about the cell cycle and how we have these checkpoints. So the checkpoints were important because that's where we check for problems that might happen in the cell, maybe problems with the chromosomes, problems within the actual cell itself. If there are too many problems that might commit and tell the cell, you need to go into apoptosis, which is programmed cell death, or, it might say if it's a problem it can fix, you need to repair that. Now, what cancer is, it's a disease when the cell makes copies of itself, replicates when it should not. So when basically cancer is we override those cell checkpoints. We keep dividing and dividing and dividing and dividing. So what happens is this cell right here, it overrid all the checkpoints and it keeps dividing and dividing. And eventually it leads to a pile up of cells because it's rapid dividing. And then it gets this mass which is called your tumor. Now normally a tumor is just a mass of cells that stays in one spot. Now if it doesn't invade surrounding tissues, it'll be called a benign tumor. Most of the time they're harmless. But as time goes on, this tumor gets bigger and bigger and can advance to what we get to the malignant stage. Now, the malignant stage is when the tumor has started to invade the surrounding tissues. Eventually, they have to get their own blood supply because they need to get the nutrition, and they can be quite harmful when they get to this point. Another thing when they get to this malignant stage is they can go through the process of metastasis. So this is when cells from this tumor break off and then go travel to a different part of the body and start a new tumor at a new location. So it's like you don't want to get to this part right here. So comparing some of the differences, the different qualities of a benign malignant tumor, you can see a lot, like when it comes to structure. Early on, the benign is going to resemble cells in that area. But as the tumor goes through more and more division, you're going to start to see the cells look more erratic. Growth, the longer it is there, the faster it's going to grow. It's going to start to get very invasive. This is just showing you some of the differences. So let's look at some pictures. So this is showing you some normal cells versus some cancer cells. So most of the time when they are doing detectors of tumor, they'll probably do a biopsy, and then they're going to look at these cells with a microscope. And what they're looking for some hallmarks for cancer. So one of the things is looking at the nucleus. So most of the time it's going to be that small little dot right there. Cancer cells, it's very, very large. They're going to look at how many cells are actually going through dividing or committing through the interface. Remember, most of the cells spend 90% of their time in interface. Well, cancer cells, they don't. They are constantly dividing. So if you're seeing a lot of cells undergoing mitosis, that's an indication, okay, maybe that's cancer. They look for abnormal shape and size. So you know what the normal anatomy of the cell is going to look like for that type of cell. Cancer cells are going to be all kinds of different things happening. They're going to be very irregular. And loss of normal features. So we know what a cell, depending on the type of cell, is going to look like. If it looks totally erratic, it's a good indication it's going to be a cancer cell. So it seems that everything today causes cancer. This is one of your assignments where you actually look for, some kind of thing that causes cancer, a short little paper. I swear on the news at least once a week, it's like they find a study that such and such has shown to lead to an increase in cancer. Now there's two different ways that you can get cancer. One is through genetic. It does come down to genes, and one is the BRCA1 gene associated with breast cancer, and it has been shown to run in the family, so it can be passed down between different generations. So if it has been shown in your family, one thing that people do is they get genetically tested to see if they have the good version or the bad version. And if you have the bad version, maybe you can do some preventative care, before the process of cancer actually gets to that stage. Now, environmental is the big one. This is the one where that probably comes across most of the time on the news. So this is when we have exposure to carcinogens, which causes those mutations that lead to cancer. The big one, smoking, has been really shown to lead to cancer. UV light exposure, so wear your sunscreen. That's a big one. I've seen all kinds of different things. I've heard of diet, um, how we're, I've saw some studies how we're attached to our computers has led to that too. For a while, I remember when people are saying cell phones are causing brain cancer. It seems every day there's something that leads to cancer. So that's one of those assignments. Maybe you'll find something interesting. It's kind of interesting that pharmaceutical drugs are here. So they have this drug to find kind of cure a problem, but then eventually they find out it causes cancer. So it's kind of stressful, but see, I like to always see what y'all have to say and what weird things you find. It's like, oh, if you eat more red meat, you're 2 times likely. It's interesting to see what the vaping stuff might do, because that's such a new thing, and we're starting to find some risk with that. So have fun with that assignment. All right, so there's two ways you can get it. There's the environment, and then there's the genetic. So this is going to talk about the genetic part of it right there. And there's two different types of genes. There's going to be oncogenes and then tumor suppressor genes. Now, oncogenes, when they're normal and happy, they're going to be called proto-oncogenes. So these are cells that normally help your cell grow. Now, they kind of have like a Jekyll and Hyde thing. So they're normal and happy, they're called proto-oncogenes. But when a mutation happens, it's going to change it. It's going to become the bad gene. And when it becomes the bad gene, it's permanently bad. It can't go back. So when this happens, it is turned into what's called an oncogene. So normally, the proto-oncogene, good happy one, is going to help regulate the cell cycle. But things happened, he became mutated, became the bad version, which is the oncogene. And once it's activated into oncogene, it's, a hallmark for cancer. So I kind of use this analogy right there. Think of it, the cell's like a car. So you have to have a way to control how fast you can go. So think about your gas pedal. Sometimes the cell, you have to kind of nudge it along. Okay, we need to speed up a little bit. So you kind of step on the gas, but you can release it. You can take your foot off the gas pedal and, it'll slow down a little bit. So think of a proto-oncogene as like a functioning gas pedal that you can push it down, it'll go up, but if you take your foot off, it slows down a little. Now, when it becomes the oncogene or the bad version, it is when the gas pedal is stuck down. So it's all the way down to the floor. You can't slow it down. It's just going to keep dividing and dividing. Now, tumor suppressor gene is like the opposite. So normal, they slow down cell division. So they're kind of like the brake pedal, if you want to think of the car analogy. They're going to repair your mistakes, tell your cells like, okay, you can't fix that. You're going to have to go through apoptosis. We want these genes to work properly. Now, when they don't slow down the cell, they pretty much become inactivated. The cell can grow out of control, which leads to cancer. So these are like your brake pedal on the car. Normally, it's going to slow you down. So it's like, okay, we're going too fast, so you step on the brake a little. Now, when something goes wrong with the brake, so when the brake line gets cut, no matter how much you try to activate it, it's not going to work. So when these become inactivated, your tumor suppressor genes kind of combines with the activation of the oncogenes to go through and cause cancer. Let's link it in this picture right here. So we have your normal, and you get exposed to damage all the time to your DNA, and your cell's very good at repairing it. There's environmental stuff that is exposed to you, but your cell keeps repairing and repairing and repairing. Now, sometimes you do inherit certain mutations that your cell can't overcome, and this will lead to mutations in your somatic cells. So if we can't repair the DNA, we get to this point right here where we have some mutations. It can lead to where we have impaired apoptosis. So apoptosis is programmed cell death. So that's basically, if you can't stop cell death, it's just going to stay alive forever. You can activate the growth promoting oncogenes. So this is where the gas pedal is pressed down. Inactivation of the tumor suppressor genes, so when your brake pedal doesn't work. When you have combinations of all this, we start to get the cancer. So this is why cancer is like super stressful, because people are like, we spend so much money researching it. How can we have not figured out how to stop it? Well, the causes is numerous. There's so many different ways that it can lead to and be caused by. And that could probably be why it's very hard to find certain treatments for it too. So one of the biggest treatments is just to remove the tumor. And the goal is to remove the entire tumor. You know, if you can remove it, then you can probably prevent it from going to metastasis. Now, you want to remove as much as possible, but sometimes you can use it debulk, so you can remove a bunch of it. But if it's starting to invade like healthy essential organs or tissues, you might leave that part behind and then use that in combination with different treatments like chemo or radiation. Sometimes it's just to ease some of the symptoms, depending on where the tumor is, might be causing pain. I've seen some examples where they use cryotherapy, especially when it comes to treating skin cancer. This is just a little bit more different ways that cancer can be treated. I've seen a lot about the use of lasers. They're going to use this to cut through tissue, especially when it's really precise. You know, it's great how our technology has increased over the years. I've seen it where they've used a hyperthermia, where they expose it to high temperature and use heat to possibly kill the cancer cells, but you got to be careful that you're only targeting just a cancer, because high heat will kill healthy cells too. Also photodynamic therapy, where you're using light to possibly kill the cancer cells. The one that you're probably used to hearing about is either chemo or radiation. Now, what the radiation does is it kills cancer cells or slows their growth by damaging their DNA. So they already have damaged DNA, but we're going to damage it even further. And our goal is to damage it to where they can't repair themselves and they start to stop dividing and die. So we already know it's a messed up cell, so we're going to mess it up even more to the point where it can't fix itself. And when the cells die because they're damaged, then the body breaks them down. Now, with radiation, it doesn't kill them right away. It takes time, several treatments to officially, damage it enough. And one thing it does affect nearby cells is just a link to talk about some of the side effects associated with radiation. Probably the big one you probably think about is chemotherapy. So this is using drugs to kind of stop or slow the growth of cancer cells. Now, what chemotherapy does is it's going to target fast-growing cancer cells. So cancer cells, they have the hallmark of having a rapid cell cycle. Now, there are some cells in your body that's just the natural way they are. They naturally divide really fast. Well, chemotherapy can't determine between healthy cells and unhealthy cells that divide rapidly. So example, cells that line your mouth and your intestines and your hair are rapidly dividing. So when the chemo is applied, it's still going to apply to any cell that's going to be dividing rapidly. So your healthy cells that have that hallmark are targeted too. So that's why you see some of the different side effects. And there's all kinds of different ways I've read about using immunotherapy, so let's help use the immune therapy to attack cancer. Also using hormones and stem cells. So there's a lot out there, and like I said, there's not one exact cure-all, but we're making advances. So here are some videos that review the cell cycle and a little bit about cancer. So the next video is going to cover mitosis.